Okay. So the next question here is the number of fragments generated when the peptide. So this is the peptide sequence given, the whole sequence is subjected to the complete digestion with trypsin. So here it has been asked that how many fragments will be generated after this polypeptide is it digested with the trypsin enzyme. So trypsin is a protein which is an enzyme which works as endopeptidase. So there are, okay, so there is one exopeptidase, exopeptidase and endopeptidase. Okay, so for exopeptidase, that means if the polypeptide chain is being cleaved off not internally from the external. So suppose this is a polypeptide chain. So if the digestion or cleavage of the polypeptide is happening from the outside, that comes under the category of exopeptidase. For example, carboxypeptidase. Carboxypeptidase is an example of exopeptidase, and endopeptidase where an internal cleavage site can be targeted. So trypsin is one of the endopeptidase, and also we need to understand the residues. So for any enzyme activity, it should fit in a particular location in the substrate. So that substrate is known as the active site. So the active site where trypsin fits and perform its action, or the residues which is recognized by trypsin to perform its action, should be known. Okay. So for trypsin, I would say the recognition residues are lysine and lysine and arginine. Okay. So lysine and arginine are the recognition residues for trypsin digestion. So trypsin cleaves the carboxy end of lysine and arginine. Given that, okay, so let me show you. Yeah. So uh, this is one amino acid residue. This is another amino acid residues. So here, if in this, suppose this is N minus 1 amino acid, this is N amino acid. So this positioning is of for the amino acid. This labeling is for amino acid. Okay. So if N minus 1 is lysine or arginine, the trypsin would digest the carboxy end of of the lysine or arginine residues, except if N is not proline. If N is proline, or this particular R group is substituted with the side chain of proline, then trypsin digestion will not take place even though lysine and arginine residues are present at that position. Okay. So let's see here in the structure. So we have the structure. This is A, N, D, C, Q. So you should know the one letter code for the amino acids. So for lysine, the one letter code is K. For arginine, it is R. or sorry. So for lysine it is K, for arginine it is R, for proline it's P. Okay, so there are different amino acids. So you should be knowing the one letter code for these amino acids. So here if we try to see, let's see how many lysine and arginine residues are present. So if we'll see, so there is this one, lysine, another lysine, arginine, another arginine. Okay, so there are two lysine and two arginine residues are present. So if trypsin will perform its action on all the four sides, then total 1, 2, 3, 4, 5 fragments would be generated. But we should also keep in our mind that the trypsin would not work if the adjacent or the next residue of lysine or arginine is proline. So here in the first lysine, after lysine it's F, which is phenylalanine. So here the trypsin will digest or cleave. Then the next lysine, the adjacent amino acid is P, that is proline. So due to the presence of proline, here the trypsin action would not work. Let's see here the arginine, after arginine Y, that is tyrosine residue is present. So here trypsin can cleave or digest this polypeptide. Okay, and here in the last arginine residue, after arginine again proline is present. So here also trypsin action will not work. So there are only these one in two places where trypsin can cleave the polypeptide. So after these two cleavage, the polypeptide will be divided into 1, 2 and 3. Total 3 fragments will be generated. So the answer would be the number of fragments generated with this peptide is subjected to complete digestion with trypsin are 3. So the answer would be 3. So for cracking these kind of question, you should be knowing the action, the specific residues where these different enzymes cuts the DNA, cuts the protein. So here is a list of the specificity of some common endopeptidase. So trypsin, which we have already discussed. So trypsin, it states that C-terminal site of positively charged residue, arginine and lysine, or Rn-1 are positively charged, but Rn should not be proline, which I have just now told. Here, if this is N-1 and this is N amino acid, so N-1 should be a positively charged amino acid, or you can say arginine or lysine, and given that N should not be proline, and the cleavage action is at the C terminal. Okay, so suppose there are if I'll make a chain here, polypeptide chain. So this is one. So this is the polypeptide bond, which is CONH. It comes the next carbon. Okay, so here let's see if I'll see this one. I'll see this residue. Okay, so if this is the n-1 carbon, it is saying that C terminal side of the positively charged. So here this is the C terminal side, this one, and this is the N terminal side. So here for the trypsin, the action is at the C terminal site here. Given n-1 is lysine or arginine. And next preceding should not be proline. In case of chymotrypsin, R n-1 should be bulky hydrophobic amino acid residue, or we can say the aromatic amino acid residues that includes phenylalanine, tryptophan and tyrosine, and Rn must not be proline. Okay, here also this condition applies. So here also Rn should not be proline. Okay, then next comes elastate. There Rn-1 should be small neutral residues, uh, for example alanine, glycine, serine, valine, and Rn must not be proline. Next comes pepsine. So N-terminal site of Rn. So that includes leucine, phenylalanine, tryptophan or tyrosine. So pepsine cleaves at the N-terminal side of these listed amino acids. So pepsine action will be towards the N-terminal side. So if this is the, okay, or we can also say Rn is leucine, phenylalanine, tryptophan or tyrosine. And here also Rn-1 must not be proline. Okay. Thermolysine again N-terminal side of hydrophobic amino acid residue excluding tryptophan. And Rn-1 must not be proline. Okay. So these are the few endopeptides and their specificity. So the question which we have solved, so if you remember the cutting site or the specific residues where endopeptides make a cut, and the conditions where the cut will not happen, like for the exception of the proline, presence of proline in the adjacent as the adjacent amino acid residue, the enzymes will not work. So these things, keeping these things in mind, these type of questions can be easily solved. And also you need to remember the single word coding as well for the different amino acids. Okay, so this list should be remembered and the questions like this can be solved. And this is my NPTEL reference where you can study about the enzymes in detail. Thank you.